This is the component 2 Taskbook, Continuity of Life, and we're up to question 9, Genetic Engineering in Applications. And E. coli produces an enzyme, and that can break a strand of DNA, and it'll always break in the same place, a recognition sequence. Now the diagram below shows some human DNA, and part of the sequence is labelled and pretty straightforward to start with. Write the base sequence, so you're just using complementary base pairing here, so that will be C to G, A to T, T, A, and G. There we are. And this C, T, T, A, G. There we are. So pretty straightforward. Draw arrows on the diagram to show the points where the enzyme would break strands 1 and 2. So you are looking to find the information where the break always occurs where guanine is adjacent to adenine. So where guanine is adjacent to adenine, so guanine adjacent to adenine, guanine adjacent to adenine, there's one there, and there's one there. So that's where the cuts are taking place, and we've drawn arrows and we've answered the question. In the space below, draw the separated piece of double-stranded DNA. Well, if you can imagine this is a cut, so this would cut here, that would cut down there, that would cut, and then you've got that there. So the piece that we've got is kind of this piece here. So that's the piece that we need to pop in. So we just need to draw it exactly like that is there. So we've got A, T, T, C, and then none of that. So we just got the G down there because that's where these are where the cuts were. We've got a gap, and then a G with a C, T, T, A. And when it asks you to draw a diagram, always follow the style of any diagrams that have been given in the question. So I'm just going to make this look rather scruffily like what we've got. And it doesn't matter if it's scruffy, it just matters whether it's right. Okay, and that's what the diagram looks like. And that's the bit that we've got. What is the most obvious feature of the sequence and bases? A strange question, obvious. Well, if it's not obvious, then it's not obvious. This is palindromic. So the bases there, look at the C, T, T, A. So in kind of reverse order on each side. So bases are in reverse order on each side. Obvious to the examiner, might not be quite obvious to everybody else. After the reaction, what is named given to region X? So region X, it kind of, on the diagram it kind of says it's there, but this, if you think about it, it says region X there, but if you look at it from this side, it's probably a bit more bit more telling. We've got exposed bases that are capable of forming hydrogen bonds with their complementary pairs. So the name for that, this is our sticky end. If it was just a straight cut, then that would be a blunt end, but as we've got exposed bases, that's now a sticky end. And name the group of enzymes to which EcoRI belongs. Well, this is the enzymes that cut, enzymes that break DNA, and they've all got a name. So they are restriction enzymes or restriction endonucleases. I'm going to put the full word. Restriction endonucleases. That's the family. And then we have genetic engineering makes use of the above technique to open circular DNA in a bacterium, and then we find a human gene and somehow put it into this bacterial DNA. Name this bacterial structure, so the small loop of DNA. That should be okay if you know your bacterial structure. Plasmid. The enzyme used to fix the fragment into the bacterial structure. So that is the enzyme that on this diagram, where we've got a cut there, we'll make the joins here. That will ligate this together. So the joining or ligating is ligase, or DNA ligase. And there's another enzyme, the fourth enzyme in genetic engineering, where we can make our DNA from messenger RNA. And if you remember, with protein synthesis, the messenger RNA is leaving the nucleus and it's been read to make a protein, but it's a copy of the gene made from DNA. And this is the other way around, so we're making the DNA from messenger RNA. So the normal one is transcription. This is reverse transcription, so it's going the other way. And so the enzyme is reverse transcriptase. Reverse transcriptase. And the other enzyme of our four in genetic engineering, our four main ones, are making DNA, so making it from the single strand to a double strand, which is adding nucleotides on, which is the same one that's used in DNA replication. So making DNA, and that's DNA polymerase, which makes the DNA polymer. So you've had all four enzymes, so you should know what each of those enzymes does. The advantage of this technique over the method shown in the diagram, well, here we've obtained this DNA just from a normal cell, which contains all of the genes in a human genome. Whereas if we use reverse transcriptase, we can find a cell which produces the protein that we want. So it's producing the protein, so presumably that would have messenger RNA floating around in the cytoplasm for that protein. So it's a way of just finding that particular messenger RNA for that particular protein, rather than having to find it in the whole mass of genes. So it's a way, these cells will only contain the gene that is required, floating around the cytoplasm in the copy of messenger RNA. So easier to find the required gene. Easier to find the required gene. So there's not lots of other gene copies on messenger RNA floating around because this cell makes that protein. Other things you could possibly say, the messenger RNA has got the introns removed, so it's much easier to use. It's just got the exons. So we could we've got our mark there, so we don't need to say much else. But we could say, no introns on the gene as an alternative there. Name a major international research name. So what's the name of it? And give us a suggestion how that's going to be helpful. So the name of it is the Human Genome Project. I could possibly say the 100K variant of that, but this question probably predates that. So Human Genome Project, and that was finding the complete base sequence. So we can identify lots of other things from that. So identifies the complete base sequence. And now, if you know that, you can identify other genes and so on. So 13 marks, but there was quite a lot of basic kind of definitions in there that you should have known.